Northern Plains, and then the University of New Mexico Metal Superfund Research Center, of which Johnny Lewis directed the center for many years, but as of about 2 years ago, I have become the director. And of course, I would like to thank first our sponsors, right? The people that support this work through the National Institute of Environmental Health Sciences Superfund Research Program. Also, some part of this work has been supported through the Center for Water and the Environment, which is supported by the National Science Foundation. Also, National Science Foundation Career Award, which really propelled a lot of the things that I'll show you today. And then my alma mater, Virginia Tech, who also offered me some support with microscopy analysis of things that I will be showing. And as you said, Ben, the Stanford synchrotron radiation light source through Slack, okay? So, and my research group. So let me start by just saying that, let's frame it, I always make the joke, right? That I tell my students, they know when they hear me, I need an objective that is measurable and feasible. And I also like when people frame things in a research question style. So I did that. So the overall objective and vision of everything that I will show you today is to identify, especially reactivity of metal mixtures, okay? And these are complex reactions in mine waste in native communities, as you mentioned, Ben. And in order to do this, we just have to add every tool that we know and see what we can, right? Because again, the complexity does demand us to integrate several techniques and approaches. And at the end, what we want is to answer the research question. So what are the mechanisms affecting the metal mobility in these mine waste? And I got us this morning, had a great meeting with some of the PhD students and postdocs, and we were commenting that at the end of the day, what I hope is that any data that we generate can inform both risk assessment and risk reduction strategies for remediation, right? So that's where we would like this to go. So the big picture and context of where we work, I'm showing here two sides. The first side, I'm gonna have to get used to this. But the first side here to the right of me, left of you, I always get confused with these two, shows a location of our partner community in Lugatachi, Navajo Nation. What you see up there is a site called Claim 28 site, so it was reclaimed and the waste of the area was aggregated in this site. But you can also see here where some of the houses of our partners are located, right just in front of this site. And the other side that I'll show you here in this vast land, which is about 45 minutes from Albuquerque, New Mexico, is the Jackpile mine, which is located in the Pueblo of Laguna. And I'll talk about both of these sites as examples of many sites in the United States where there is, there are literally, depending on how you count them, it's going to be even higher than what I'm saying here, but thousands of abandoned mine waste. I'm talking about the Western US simply because I live there, but as some of you who live here in the East Coast know, that mining is not unique to the East Coast, right? So, or the West Coast, it's almost, I wanna say, a very comprehensive problem, not only in this country either, right? It's in the world, but particularly we have been working with these rural communities, our Native American partners who live in the proximity of these sites. And we have over the years developed relationships and listened to their concerns where they have indicated that as you have seen from the Oppenheimer movie, which won the Oscars and a lot of visibility, but our partners tell us, right, that movie just doesn't show at all. It shows a very romantic thing of the nuclear era, but it really doesn't show the impacts or the effects that this had and a toll it has taken in the communities, right? And that's what I would like to talk today. So we need to understand then these reactions, this fundamental knowledge so that we can inform these risk assessment and remediation strategies. So another comment that I hear from people is, Jose, why are you working in these old problems? Because we are a civilized society, we have advanced so much. Why are you working on old stuff? You should work on new stuff, PFAS or whatever it is, right? Well, what I do have to say is that, especially as you saw how we struggled placing the technology here today, we are pretty much addicted to cell phones and computers and now electric cars and renewable energy and blah and blah. So what it will take is more mining and more extraction, of course, to sustain these things that we like. So I think that it's important to understand these lessons from the legacy of mining on developments for the future. And these are some examples. I actually was in another seminar that you gave me, you're right, about lithium extractions. And I've been in other talks about rare earth elements, right, that we definitely will need for the development of future technologies. So these are all projects that are coming, and it's important to reflect on what has happened in the past, so hopefully we can do it better in the future, right? That's, I think, a vision that I want to leave you here. And also was talking to Anna earlier, right, about the fact that as a director of the Superfund Center right now, I feel that with these I'm gonna say conversations and initiatives that have to do with resource extraction and evaluating how we're going to develop this as a country, then in that importance on critical minerals, that these issues require us with more urgency, all of the tools, all of the infrastructure that has been developed in programs like yours here in Columbia University and ours in the University of New Mexico. So I think that these are pressing issues that we have to keep in mind, and independent of what you want to call things, right? We have to be very mindful about the central issues and how we're going to go about ensuring that our communities are protected, right? Because environmental health is still an issue that we will have to be present into the future. And in our way of working and engaging with communities, we have been humbled many times of how awful I present and how badly I can communicate science in a way that is not understood or appreciated by our communities to make something useful out of it. So I have been very humble, I tell you, too many times. And with the support of some of my colleagues that I want to mention, Mallory Kotowski being one of them, many other colleagues, Thomas Dupree would be another one, Chris Shuey, my collaborator from the Southwest Research and Information Center, just to name a few, right? Now I'm working with another very energizing colleague, Aziz Lopez, and Anjali Molchandani, also a colleague that is here with me in a picture. We all go to the field and try to have listening sessions with our community partners. But you see this word listening. It's not imposing or giving them an opinion, because I was asked today, what do you do, Jose, when you have the science? Do you stay in a side of advocacy, or do you stay in a side of being neutral and not doing anything? And at the end of the day, what I hope is that as a scientist, even if I die today, I hope that the science we generated is useful, right? I mean, that's really the hope. So in order to make it useful, then you need to communicate it in a way that can be understood and can be appreciated. So we have done different efforts, as you can see, with artwork. And this is an example of Mallory here just showing even biomedical projects, right? Where you have to explain DNA damage, DNA repair, right? In a way with art where our partners can relate to, and we still receive feedback. One thing that I'm saying, this is the way that everybody has to do it. We're still learning, okay? So just stay there. But these are, some of the approaches that we're trying to use as we continue to develop these partnerships. But definitely, I want to embrace that none of the work or none of the data that I would show you today would be possible if we didn't have these relationships. Okay, so that's a very important thing to leave very clear. And now I'll show you some data. I just use this, my friends, for simplicity. I'm sorry, this is not the whole data that I have, but for simplicity, because of time, I am going to use these two examples of these sites. That is a compendium of some papers that are research group has published over the years. And I'll also share that when I started this work 12 years ago, you see, I was approached by some of my colleagues and asked me, Jose, can you come and work with us? But we have been, when Johnny Lewis at the time had done this wonderful job with community environmental health program, mostly on environmental health, but I am an environmental engineer, environmental scientist, and I was asked, Jose, can we get some water samples or some water analysis? So I did. And when I got this data at the beginning, I really was appalled because as you can see, sorry guys, I'm struggling with the order of this thing. I sometimes just prefer to pick it from the computer, but as you can see, the levels of uranium can go to hundreds of micrograms per liter. And as a reference then, the maximum contaminant level is 30 micrograms per liter for uranium. Some of our recent sampling, we have measured a site in the North Pit of close to the Jackwell mine, where we have measured over 1,000 micrograms per liter, just so you have an idea, right? And also, to make it even more interesting here and more challenging for us, right? Uranium doesn't occur alone, then you have arsenic, you have vanadium, you have other co-occurring elements. And in the case of arsenic, which is also, it has a maximum contaminant level of 10, there are some instances where it is exceeded, right? And the other thing is, some of these occur at acidic pHs with acid mine drainage, right? So there's that part. And also sometimes you have to deal with well-buffered waters at pH 8 or whatever, right? In that area, but closer to 9, I'd say there. Just so you see the range of complexity that we have to deal with. And this is then something that serves as a framework to set a context for the issue that we have to understand. Where does this uranium and arsenic come from? How does it occur? And most importantly then, how can we think as people that would like to reduce this risk, right, to remediate? So let me talk first about some of the work that we have done in the Blue Gap Tachi community in Arizona. And Anirban and I were talking earlier, and Catherine too, about the complexity, let's say, of different forms of uranium. If you want to get more intricate, let's say, about how there's some different species, different things, right? And it all comes down to the fact that you don't have to be a scientist even, or an engineer or whatever, right, or a hydrologist. If you go to this site, which we have been and taking a picture from up there, you can see, standing from the mesa, which is the picture that I have here below. And struggling with this thing. So you can see this picture here in my right. If you stand at the top of the mesa, where does the water go when it flows? Into this pond where all the sheep eat, right? So that's a very well-known thing, especially if you go to Arizona, New Mexico in June, where it's very hot. You will see we don't even have any water. So this is the only water that is sometimes available for these animals, right? Just to have this awareness. And also, when we zoom in to look at the mineralogy of the mine waste itself, and if we integrate scanning electron microscopy and energy dispersive X-ray spectroscopy to do elemental analysis, what we find here very clearly, especially here we are with all of these people that love geology, so it's very nice to be here, but they know that we have these bigger phases, right, that in my mineralogy colleagues call them primary, because they serve as hosts, right? You were talking about aluminosilicates, calcium, magnesium, carbonates, whatever it is, but just on top of it, you see I'm gonna call it a little brighter area, like a coating of these minerals, that they are bright for two reasons. First reason is because of the concentration. Second reason is because, for those of you who don't like chemistry, I'll explain, uranium is a heavy element. So its atomic weight is 238 grams per mole. And compared to other elements where it's like hydrogen, the atomic element is one. Imagine poor hydrogen, right? It's in disadvantage with uranium. But because it's so dense then and so heavy, it is very convenient because it shines for us very conveniently. So we can easily detect it. And when we zoom in, we can see if we identify that green is vanadium and red is uranium. What does green and red give us? Anybody, you remember when you combine green and red?
Brown.
No, you're close though. Yeah. Yeah, orange or something like that, right? So what we see here is this very nice combination. So you see the red and green, and if you bring this together, then you have this yellow. This is a uranium vanadate phase, and that is confirmed by the peaks that we see for uranium and vanadium. And we were earlier talking about lithium as we were talking. So, but Catherine was saying, well, potassium, sodium, lithium. We were just talking. Those are all atoms that will substitute into any of these structures, right? So just to illustrate the complexity of metal mixtures, right, in these types of samples. The other thing that is important here to highlight, if you go to New Mexico, especially between these months of February to April or Arizona, anywhere in the Southwest where we live, we get a lot of wind, like it was windy yesterday too here, by the way. I remember that. Yeah, exactly. But so it's so windy there that we get a suspension of particulate matter, and we get what people call sandstorms. I don't know if you've ever experienced one of those. But you just literally, you can taste all that dirt in your mouth very clearly. And my community members have always asked me, Jose, is that a concern if this material gets suspended from our mining legacy, right? That remains. And well, all I can tell you is if we zoom into, these are some boulders of rocks that are close to the miners, but you can see these fringes of these yellow minerals here. So these yellow minerals are typical oxidized uranium minerals, right? And that's why after processing of uranium in plants, the whole goal is to extract a lot of that oxidized uranium, and they call it yellow cake, right? Which is a lot of uranium oxides. But here they are naturally occurring. These are mine-based, these are not processed. And what you can see here is very clearly some of the edges of these rocks have these fringes with yellow. And when we zoom in, so you see that some of these rocks I can see even with my eyes. This is a paper towel to give you a sense of scale. But some of these are so small that I cannot even see anymore. So we take this to a transmission electron microscope in collaboration with one of my first PhD students, Sumanta Lazarala, and my good collaborator, Adrian Burley. And we have seen these clusters of carnotite nanoparticles. And I say cluster simply because you can see a lot of different minerals clustered together. This is a 0.1 micron scale, but you can zoom even more to 100 nanometers and see some like this, right? Where if the definition of a nanoparticle is that it has to be less than 100 nanometers in one of its dimensions, you can see very clearly that there's one here that is detached, just to give you an example. And when we do this elemental analysis, we find also consistency of uranium, vanadium abundance, okay? So with a lot of this, we have replicated analysis and have found the abundance of this uranyl vanadate mineral called carnotite because it's a potassium uranyl vanadate, okay? So these nanoparticles then are present. We really at this point would love to understand, right, the dispersion and how this moves, but we have definitely identified, and also with the collaboration of, let me see, of some of my colleagues, we have done some modeling. I'm not going to bore you with differential equations. I could, but I'm not going to do that today. But all I can tell you is that we did some column experiments where we packed the materials with this mine list from the community of Lugaptachi in the Claim 28 site. And what we found was that if we reacted this with both solutions that had bicarbonate and also with acidic solutions at pH 3, we found a very nice correlation where you can pretty much explain the uranium release very nicely with this curve, assuming that it is controlled by the kinetic dissolution of carnotite, of this uranium vanadate, which is very nice. And of course, and it's sometimes nice when these experiments and models coincide, but often they don't, right? We were just discussing. So this is one part where it doesn't, because vanadium, as you can imagine, yeah, maybe a part occurs with uranium, which is what this shows, but there's another part that occurs likely as vanadate oxides, right, that are not necessarily with uranium. So you see a higher release here, but the uranium curve is very nicely represented by the release of carnotite. So this suggests this important contribution, right? That if you want to understand then how that uranium will be moving in this side, you have to consider that that carnotite dissolution will be an important mechanism, okay? And then, and thanks to Peter Ligner, who is one of the developers of this reactive transport software that we use with Lotron. So we ended up publishing these papers, and there's this other very nice paper in the middle here just showing an example of how our biomedical projects and environmental projects come together, right? So this is a collaboration with Katie Sikowski, who is now faculty at UNM, and Matthew Campen, my collaborator too, with many of us who were part of this study. And what we found was that respirable uranyl vanadate containing particulate matter can exhibit potentiated cardiopulmonary toxicity in animal studies. Okay, so these are just examples of how we're trying to combine our skills together to better understand. However, I admit that this is a field that still needs, you know, a lot of good work, I must say. Okay, so and let me show you now some of the work that we have done in the Pueblo of Laguna. So this is again 45 minutes approximately from Albuquerque, New Mexico. Back in the 60s, this used to be one of the largest open pit uranium mines in the world. And what is interesting is you have the mine, and very close to the mine, you see our villages, right, of our partner communities, and the villages that are very close, Laguna, which I was very close to the feast day some days ago, and also Mesira and Paguari, right, was the jack pile mine is close to the Rio Paguari. And because of that, and this has been listed as part of the EPA National Priorities List for cleanup since 2013. And this is a map here just to show you that there's this, you have to imagine that this is a stream here, that it's an intermittent stream, by the way, because we have very unique semi-arid climates, right? Where we have sometimes very arid, no precipitation like we will have this year. We haven't had a lot of snow. So I am waiting for a pretty hot summer. And when that happens, then we also will have a monsoon season usually around July and September, and I'll show you how that plays a role in the concentrations of uranium. But when we want to communicate this data to our partner communities, then with the collaboration of Chris Shuey from the Southwest Research and Information Center, we have he has developed this very nice stoplight approach with the communities, where if it's red, it's higher than 100 micrograms per liter, definitely, you know, we should be careful with this water. And as you can expect, it's just across the mine, right? So the other part would be, it's in yellow because it's from the maximum contaminant limit to 100. These are arbitrary numbers in some ways, right? But this is what we say, okay, it exceeds the standard, and it usually, it's observed just downstream of the mine, up to this wetland area that I'll show you in a little bit why it is important to us. Because what happened is there was a dam that the community had built back in the 70s to hopefully have some water to rely when it's dry. But unfortunately, it's located in this area where we know is, elevated uranium, so it's not used for those purposes. But because that dam is there, it retains water, and then we have a little wetland that has grown, which I'll share with you in a little bit, okay? And then the areas where, of course, upstream and downstream of it, where the concentrations are less than the maximum contaminant level, right? Now, this is a very nice seasonality link that I'm gonna give the credit to both Sherry DeVore when she was doing her master's in my group. She's now a faculty at the University of Arizona. And also Johanna Blake, who is in USGS, a research scientist. So when we worked together in my group, we actually went sampling. And here are some uranium concentrations from 2014 to 2016. But we actually, Sherry had the idea, there was a gauge station of the USGS that operated from the late 70s to the 90s. So she said, I'm gonna plot the data too. And to our surprise, what we found is that the areas where you have high flow, so in the y-axis here, you can see discharge in cubic feet per second, and then the month of the year. Again, this data that you see in the graph to your lower left for you guys who are watching the screen, you can see where you have the high points. The highest flows are, of course, in the monsoon season, right? Where I just mentioned from July to September-ish maybe. But we also can have some years where you see these peaks in February, March, that is snow month, okay? When we have snow, like this year we didn't have very much. And that coincides when we saw some concentrations of uranium here in January, probably caused by that snow melt that we're talking about. And again, the highest concentrations when we had this monsoon season, okay? So, I'm dyslexic, guys, I'm sorry. So you'll see that I go back and forth with these things. But I also want to show you then that when we take the mine waste from the jack pile mine, in contrast to what we saw in Arizona, right, in the Blue Gatache community, we have all these uranium vanadates. Here we have a different story. And the story has to do with, you see these dark spots here, those are uranium minerals. But this is a potassium uranium phosphate, not vanadate anymore. This is a phosphate. And what's interesting is that that phosphate is surrounded by all of this gunk, I call it. And when we look at this elemental map here, you can see even more clearly that this gunk, if you look at carbon here, is all over the place. So this is all carbon, likely organic carbon. That, as we were talking earlier with Anirban, of volcanic influenced areas, right? So this is a sandstone formation where the mineralized deposits are rich in organic matter because that organic matter in some way serves as a glue, okay, to keep together all of these uranium minerals. But what is fascinating of this is that we see contents of uranium in 1 to 5% in the solids, meaning then if I multiply this times 10,000, we're talking 10,000 to 50,000 milligrams per kilogram. And it's a lot of uranium, by the way. But it coincides with 13 to 40% carbon in the solids. Okay, so this just shows us that the uranium is encapsulated by this carbon. And another very cool thing to me, at least, is that most of my postdoc, as I was discussing with Ben Bostic earlier, I did some work in Rifle, Colorado, because there was a project of the Department of Energy at the time there. And I recall that sometimes whenever we were able to look at reduced uranium, we had to be super careful because it readily oxidized, at least the ones that we got from the field. So it was a whole operation. We had to put things in a glow box immediately and all of that. But what was fascinating of these samples is that I got them as a poor assistant professor the first time that I got this. And we didn't even have the Superfund Center at this time, so we literally told the community to help us sample. We got indicated where, got some containers, I guess, and we still were able to see this reduced uranium. It's a uranium silicate, so that the organic matter doesn't only or encapsulate that uranium, but keep some of it in its reduced state. So one of my former PhD students, Carmen Velasco, published this very nice paper just showing how that uranium-4 reduced uranium and uranium-6 oxidized is still present, and that no matter if we have different pH reactions, we saw some changes of organic functional groups, as some of you can imagine, right? When you have those different pH reactions, but the uranium-4 was still present, which to me was interesting, right? So, and we have a project here that we're trying to develop with Ari Gibb, who is sitting down here. I owe him some samples. I put myself recorded there, that I'll have to get them to you, okay? But we do have to explore this more to understand that oxidation reduction dynamic of uranium, okay? Now, let me end this talk with some hope, for God's sake, right, in these times. We need positive hope. And I do want to leave us with thinking about remediation. Okay, so in the state of the science, honestly, in these approaches of at least what has been done in these sites is, as you saw, these sites are remediated. I'm going to use the word quotes, remediated by adding a non-contaminated layer of soil, of non-contaminated soil, right? Put it on the top and hope that it will contain it, right? And so what happens over many decades? Weather, right? So I mean, that's just a reality. And another thing then that I see now, a lot of discussion is, okay, why don't we enable some biological activity, these soils, right? So that they just don't float around. So now we talk about, you've probably heard this term from our collaborators and colleagues from the Environmental Protection Agency of cap and plant, right? So cap, but you plant, hopefully that stabilizes things. And I guess our research group has been looking into these questions. Okay, so if we cap and plant purposefully, how can we make use of both plants and those natural minerals more purposefully? I'm using the word purposefully. So this is the fundamental research that we have done in our labs. So let me then show you some of the ideas that we got just from going in the field and observing. You see all of that white, all of that white, I'm gonna call it material there, right? Those are calcium minerals, and that's where we got the idea of looking into calcium into more detail. Okay, so I'll show you both how that plays a role in understanding immobilization with plants and also understanding how we could use calcium-bearing minerals, hopefully for giving us ideas for remediation. So we'll get to this. So let me see, the first thing is, let me talk about the plants first, because I wanted to show this to, especially because there's some exciting stuff that's happening here in Colombia with plants. So this here is evidence that I sometimes work, actually. So this is me in the field, sampling with my good collaborator now, lecturer in Newcastle University, Lucia Rodriguez Freire. And we were sampling. And this is, sorry guys, the area of the wetland that I was, I'm getting ahead. Give me one second. The area of the wetland that I was talking about before. And what you can see there is, as we sample in the wetland, we see grasses, we see salt cedars, we'll see all of these plants. And when we sample them, we saw that, let's say, some of the sediment here is more what I call background concentration of uranium, around 3 milligrams per kilogram. But we started to see that the roots had 20 to 50 milligrams per kilogram. I'm like, okay, well, this is at least 10 times higher or more, right, than what the sediments have. But in this case, we didn't see much accumulation to the stems. So as you can imagine, right, when I show this stuff to my colleagues, they're like, Jose, but how do you know that this is not an artifact of you? So we took this to the synchrotron, right? So this is a collaboration with Juan Lezama, in Stanford University and Scott Fendorf. And what we were able to do here, this is beamline 10.3 with Sam Webb, who is amazing, automation of everything, which makes it very easy for us. We don't have to stay overnight like you and I did back in the days now. It's easier now. But what is cool of this is that we open these plants from. So I'm showing you from the inside and what the in this case micro X-ray fluorescence mapping shows is that the uranium accumulates in the outside layers of the plant. So this is like almost the epidermis and cortex of the plant. Yes. We did. We had to. Yes. Yeah. That was a whole nother conversation. I will get to you later. Yeah. We went through so many things to get this sample fixed in the right way. So these are fixed in epoxy. That's right. And but we saw that uranium very consistently. This range, as you see, I almost like to think in the skin of the plant, right? In some ways, thinking. So after that, then Elian and Hayek. So this was a salt cedar plant that I showed you earlier. And Elian and Hayek, who is now a research professor in the College of Pharmacy, had this great idea. Why don't I grow some mustards, which are easier to grow, instead of dealing with salt cedars? I don't want to work with salt cedars in that way, the same, right? But you can easily work with mustards, a hyperaccumulator plant, a Brassica juncea. And this is a little setup of what Elian set up at the time, you know, with this UV light, very nice greenhouse there. Acclimated the plants to make them happy with some nutrients, like you and I need those nutrients, like I'm gonna have a paella tonight, so something like that. And then you end up exposing these plants with calcium, and I explained to you why calcium, right? And also with uranium that is in the concentration, so it's the maximum contaminant level up to 700 micrograms per liter, which I showed you we have measured in the Rio Paguari. And what we saw then, let me just show you, after that experiment was that we saw some uranium accumulation in the roots, but only when we had no calcium, which was for us, right? Okay. And what we ended up realizing was, okay, calcium, uranium, and carbonate form these complexes that tend to be neutrally charged or negatively charged above pH 7. Okay, we're working on pH 7 here. So what we thought is, okay, if let's assume that the charge of the plant is negative, what happens with negative and negative? Repel, right? So there was not a lot of attraction. But what was interesting is that even if we didn't see a lot of accumulation, Elian noted, Jose, I still see some of these spots, and I wonder what they are. So she had the idea also, again, this is Elian's creativity. Why don't I do another experiment, Jose, but I'm going to use, instead of using 700 micrograms per liter, I'm gonna use 90 milligrams per liter, just so that I can really see that uranium and see if there's any precipitation that would happen. So what we saw was very fascinating, because we saw that, yes, again, calcium decreases uranium accumulation in the roots. But look at this. This is a very cool finding, because as the calcium increased, what we saw was that the uranium content went down, but in the shoots, we started to see more uranium. Oh, okay. So why is that? And when we looked at microscopy analysis, so this is TEM analysis, we ended up seeing this uranium precipitation with phosphate forming these needles. So you see that, yeah, you might have more uranium accumulating in the plants, but in the cells, but it is outside of the cells. It is these needles that the cells are smart to not let it come in. Those needles outside, right? Because you can think of this as membrane, right? But as soon as you have that calcium, what that calcium did is, you see these needles end up not being clustered this way, but they start not being as aggregated anymore. And they start to come into the cell. And even when you see in 6 millimolar, it's interesting, they are already inside. So this probably has to do with a mechanism of I'm gonna call it, a biochemical mechanism, right? And some colleagues now have done a phenomenal job with metagenomics and metabolomics and all this stuff, showing this very nice role of calcium pumps in plants that can facilitate not only the uptake of other cations, I'm gonna say, and anions too, right? But that is what facilitates then the input of uranium when that calcium is in. So I'm giving other people one, so you think, right? We were talking about that earlier. And also, finally, I want to leave you with this thought of the using minerals more purposefully. So I want to honor here a scientist that I really admire, John Sakara from the Pacific Northwestern National Lab that unfortunately passed away some years ago. But his group did some phenomenal work in PNNL with uranium phosphate immobilization, right? So that gave me some idea because, and Dan Jamar, also my former postdoc mentor, has done some very nice work. But I felt like, can we use some of that to also immobilize arsenic? So the question here is, can we react uranium and arsenic mixtures with calcium phosphate and maybe calcite or limestone, as I'll show you here in a little bit, to immobilize both uranium and arsenic? So these are some experiments that a postdoc of mine, Han Hua, did in collaboration with many people from my group, and also Isabel Mesa, PhD student in my group. So we have calcium around the concentrations that we have used, but also we use phosphate, inspired on these concentrations that we saw from PNNL, okay, at 2 millimolar. And we use pH 7, 8, 9, 10, 11. Again, this is a beaker reactor where you have everything mixed together. And then the duration of the experiment is 7 hours, but Han left 1 hour for each pH, okay? So what is very interesting of this experiment is that we saw that especially when you reach pH 9, you can see that both, again, so let's look, these panels, what I have here in the top is calcium and uranium, phosphorus and arsenic. So where you have in your left, it would be, you have in the top uranium and in the bottom you have arsenic. Around pH 9 is where you see both starting to come down, let's say. What is interesting is those arsenic concentrations started, pretty high and came down to micrograms per liter, closer to the maximum contaminant level. So we're very encouraged, let's say, by this result. And this was published in Environmental Science and Technology. What we found that when we looked at the reactive solids is that calcium and phosphate precipitate in this area, right, where they are commonly found. But you also see that that's where arsenic and uranium co-occur. I'm not necessarily gonna call only co-precipitation because some of those spots could be absorption too, but they definitely coincide in location. So this was a very encouraging result too.
Can you go back to say?
Yes,
One more.
one more, yes.
Because the uranium goes up.
Yeah.
Arsenic.
Yeah. The uranium goes up at the end a little bit, but it stays still in the micrograms per liter. It goes up, Steve. I'll answer that question in the Q&A if you don't mind, so that I can.
Okay.
Okay. Yeah. But again, we started with uranium and arsenic concentrations in these experiments in the milligrams per liter, just so you know. So we are micrograms per liter. So even though, yes, they go up, but they go up a little bit. But I'll explain to you why, okay? In a little bit, when I show you the columns. So we did a column experiment, now with lower concentrations. Those were in milligrams per liter. We started very high. Now we said, let's use water from the Rio Paguari, and let's spike the water with some arsenic. Unfortunately, because that day we only had arsenic at 3. So we had the idea, let's spike it with, this was with Nova Jamison actually at 200. And then, these are all the concentrations that just occur in the water at pH 8.2. But then we increased the pH to 9 and 11, just inspired on the batch experiment to see what we found. And what we were able to see here is that uranium at pH 9 and at pH 11, as you see here, it increases, but it stays still in the micrograms per liter. Why it increases is because that is complexation at pH that is higher than 9 with hydroxide. Those are uranium hydroxides that are already causing the increase at higher pH. But they still remain in the micrograms per liter below the maximum contaminant level. And what is interesting here is that the arsenic, only at pH 11, it stayed down. At pH 9, we didn't get great results here, but at pH 11, we get them in the right micrograms per liter. So this is unpublished, but we're working in this paper. This is what I was doing in the spring break. I told them. So I am really hoping that we can continue to further develop these ideas, especially because I talked to a person from EPA in New Jersey, and they were telling me, Jose, this is an exciting result because we're actually working with cadmium, and we're doing something similar with hydroxyapatite here in New Jersey, and we found something similar. Okay, well, that's a good validation, right, of being able to use hydroxyapatite more purposefully. So I did show you here that we got more than 90% removal. It's not 100%, Steve, but more than 90% removal for uranium and arsenic at pH 11. This is the cartoon that Han came up with for this. So this paper was, it received one of the 2024 papers of the year for NIHS. It does show how this reaction of limestone and with calcium and phosphate can hopefully help on this immobilization of both uranium and arsenic. And I'll just close here because I know it's late. I started a little late with this thought. So first, that the pilot studies we have done are all bench, right? We have to go and test in more realistic conditions, right? So I'm looking forward to some more pilot testing that we can do, and that's why I was excited when this colleague from EPA contacted me, because I think those types of studies are more important. And with this, I'm saying, well, if we can use plants and natural minerals more purposefully, can that lead to the development of culturally respectful approaches for remediation, right, that we could use? But here's another thing with the whole deal of critical minerals now, and I tell you this because I have received calls lately of people that are interested in mining again and talking more about, hey, can we recover uranium? Some people are always going to see it that way. And I think that those questions are important and useful, and that's why as we're thinking of extraction of critical minerals, again, all of these science that we have developed applies to that too, right? So we just have to keep an eye, and that's why I was happy to see the lithium presentation today, because that's exactly what we all have to keep an eye as these developments are taking place, okay? With that, I just want to thank a lot of people that collaborate with me, but as most importantly, our partner communities of Lugan-Tachi and the Pueblo of Laguna, our Laguna Environment Department, and then all of our colleagues from the University of New Mexico, University of Arizona, North Carolina State, New Mexico State. I have a lot of people, so Scott Fendorf and Juan Lezama from Stanford, and most recently also my colleagues from Notre Dame, Peter Burns, and from the University of Iowa, Tori Forbes, who have been super helpful in providing their expertise. So with this, I'll close it out. Okay, thank you. Yes.
There's a question in the chat. Robert Rice wants to know, is the EPA maximum contaminant level for uranium of 30 micrograms per liter sufficiently protective? Is this an old number that needs revision?
I agree. What is the name of the person, Robert?
Robert Rice.
Robert, my colleague, Johnny Lewis, is one of the biggest proponents of exactly what you're saying, that maximum contaminant level could be investigated more, if we can put it politely, right? So I think that I agree with what you're saying, and that's why I encourage funding on these areas, right? So that we can address the toxicity and what this does to health. So I agree with that, and I don't have any good answer other than saying, Robert, that those types of studies these days, let's say, we'll see where the funding goes, right?
And
Ben.
so one of the challenges in getting these apatite-based remediation or pyromorphite for lead or things to work is, you have to have the reactants in reasonably high concentrations to get them to form at any reasonable time scale. And you don't really have that when you have a soil matrix that keeps the phosphate levels low, for example. So what are some approaches that you think are useful to try to overcome sort of the kinetic limitations inherent in a natural complex system that really are hard to, so different than actually happen in the model system?
Absolutely, and my answer to that would be, you know, Ben, we're not looking for perfect, I'm not looking for 100% removal either, to be very honest with you, right? We have to start somewhere. What I would consider is developing specifically these pilot scale types of tests. One idea that I would have is consistent with what PNNL already has proposed, right? Is to have some control area where you can really monitor closely. But instead of just relying on nature to do its job, which is what we call natural attenuation, that's another conversation, but maybe you can have some seeds strategically placed, but then you would have to understand then the hydrodynamics and all of these things that have to do with the field conditions. And that's where I would love that discussion with more practitioners, right? To see how these things are done, because I know that there are pilots that are already done for some sites, but this is where the fundamental science, I hope, meets with the engineers and people that can think more strategically on how to do it on the field. But I honestly, because I haven't done it, I cannot even tell you, right, this is the way. All I can tell you is I humbly accept that we have done this in pilot, batch, in column experiments, but I'm ready to see how we could apply something like this. As it was done some pilot experiments, as we know, with the phosphate in Hanford. And again, we know that we're gonna have disadvantages, right? I know that it's not gonna be perfect, but I think we have to look for solutions, Ben, that make us progress from just adding dirt and see what happens, right? But be more purposeful about what potential reactions can you enhance. I'm gonna use this word, right? There, but I don't have any specifics because I haven't done the work at that level. But would love to get one. Yeah. Yes. Steve, okay.
So,
Yes,
couple things.
yeah.
The Aguate Reservoir, and you said there was a wetland before it, and you showed that picture where it's at, but I was surprised. I mean, wetlands are usually reducing, and yet you had very low concentrations in the wetland soils. I would have thought it would have accumulated more. So that was one question that related to that. The other is there standing water anywhere in the reservoir that's been there since the 70s? Or is it ephemeral, just like the stream is ephemeral?
Yeah, it's so ephemeral is the answer number one, because we have such a semi-arid climate, and there are years that it's so dry that you don't even see water. I mean, we have that. To your first question, I didn't show the whole story. I can send you a paper of the group on that. We have seen uranium concentrations in the wetland in sediments, not only at 3 milligrams. This was an example, but of course it's a variation, right? We have seen up to hundreds of milligrams per kilogram, and I agree with you that it probably has to do with the fact that you have more organic matter there. It probably leads to more deposition of that uranium. And yes, it's very likely there are reduced zones that I haven't looked into, but I work mostly with superficial waters, and that's why I have been focused more on the, I'm gonna call it oxidized conditions.
Yeah. And then the other question was, one of your photos had one of what looked, I thought, was a pile of old tailings.
Yeah.
But there were these horizontal lines that continued out into the more natural landscape as if they were old lake beds. And it's like, how can that be? Because it's so dry there and it's been dry for a long time. It almost looked like historical.
Yeah, well,
You know, as in paleo, thousands of years old, and that made no sense to me.
I'm sorry, man. And one of my friends in UNM would have said, well, say just to, what does it control? Didn't work. Sorry. It's because I'm working with this. I'm having some issues here, man, but I would love to see what. It would, but keep talking.
That one right there. See how those lines, maybe the pile continues, but there's those horizontal light lines that continue over to this. Yes, that's the pile, but then it continues to what might not be the pile.
Yeah, we have a combination of what I would call natural mesas here all over the place. This is where the open pit was, but it's covered with this soil.
Yeah.
That's what it is.
But what are those light horizontal lines that continue the whole way, those ones.
You know, it's hard to say, and there's a lot of extraction still of calcium minerals in the area too. So I'm not 100% sure that this would be natural or not, because I know that there are some deposits.
Is
It
there a
could be
road
road.
in the hillside?
Yeah.
Okay,
Yeah.
I was
Yeah.
just curious.
Yeah, I'll take you if you come. Let's do that.
Deal.
With,
There's another question in the
Yeah.
chat from
Yeah.
Manish Kumar saying,
yeah,
for your research, have you ever used any biomaterials or biomolecules for either recovery or as a sensor? If yes, how did you use it and what was the comparison like with conventional methods? If not, then do you have any upcoming projects in which you are planning to explore it? Thank you.
so I assume that this is Manish Kumar from the University of Texas, or it doesn't say. It's my good friend. Hi, Manish. So, yeah, so I have done some work, Manish, lately with one of my collaborators in the University of New Mexico. His name is Gabriel Lopez, actually with several, Matt Lankin, Gabriel Lopez, and Nick Carroll. And Soundapan, also from Navajo Tech, another one of my collaborators. We're trying to understand how we can use DNA aptamers and some other biomaterials I'm gonna call for sensing. Right now, that project, I would say, is in its infancy, and later from, I may just have to be honest, very far from being close to our gold standard ICP or any of these other methods, but I like the idea. And I would say that from the biological membranes materials, it would be good to explore that, but the challenge here, Manish, for remediation, as Ben Bostic was asking me, you have to imagine, and as Steve was saying, this super vast area, so this is not a point source treatment, right? You need a more passive, I'm gonna say, extensive land treatment that is affordable. So I'm not sure that point use would be super useful. They could be probably for water, but this is also a big solid waste management problem, right? So if that one I'm less certain about, but I like the idea of the sensor for sure, and we can definitely reach out to each other to see what we can do to collaborate. Without a doubt. Thank you. Yes.
So I have many questions. I'll ask you two short questions.
Cool.
One is, so the site where you have uranium generate, there, have you thought about using calcium? Because my understanding is calcium uranyl granulate is super insoluble, right? So that helps stop that, like chemical pH in the presence of calcium makes it even more insoluble. That's the first question. The second question is, what's the oxidation state of uranium in the plants that steps that use?
Yeah, so let me answer the two questions. So we started, and I believe we've done all the experiments with plants with uranium-6, all uranium, no redox. So very open to a collaboration there, because maybe there is some reduction, I have no idea, right? The other question on the use of calcium uranyl vanillase, like to uriaminate, we have seen it naturally occur, and I think it is a good idea. I definitely think it's a good idea. So yes, to do that, it would be nice to see that type of development.
Thank you so much. It's really what I love about the Superfund is the interdisciplinarity that can really help us think, okay, based on all these processes that you've studied from the earth sciences perspective, what does it mean for uranium in the human body? And I was thinking about what you mentioned about these membranes that are kind of protecting from the toxicity of the toxic uranium, and would that be happening in the body too? Like we know uranium goes into the bone, and now we understand better because of the calcium connection. And but then I was thinking a very silly idea. Have you anybody studied the kidney stones of people from this area? Has anybody done an analysis? Could you do it, and could you see the uranium in those kidney stones?
Yeah. So, Anna, this is a wonderful and challenging question that you asked me. I would reach out to Johnny and to those types of colleagues to see. I don't wanna tell you nothing has been done because I learned a lesson from Johnny. You never say that, Jose, we've done a lot. So, as you know, with Johnny, right? So, and I would have to say, we would have to check if that has been seen. I know there has been some attempts to look at this pathway of accumulation in kidneys, but I am not sure up to what extent, and I do like the idea of the kidney stones, right? If they are already forming there, I think it's a great idea if it hasn't been done, but it would be good for you to respond to Johnny
Yeah.
and also check on this and tell her that we had this conversation. So I don't know, but I love the idea. The other question that you asked, so we do know about the accumulation in bones, and I think Catherine has done with Daniel a very nice job trying to explain those pathways, right? So I really enjoyed Catherine, what you showed me when you came to New Mexico on that end. What I want to say is there's a big question, Anna, which is, okay, the water, for example, in the Rio Paguari that is at pH 7, probably because there's so much calcium and carbonates and uranium, is likely uranium calcium carbonate complex. But for our laboratory studies, we usually use uranyl acetate or some of these other salts, right? So we have been talking with our colleagues of our center, people like Julie In, Elicio Castillo, Alicia Bolt, Sebastian Medina, so these are some of our exciting biomedical researchers, Katie Sikowski too, on understanding better what is the mechanism of accumulation of this water, not of the uranyl acetate, because that's a model of this naturally occurring water. And one of the ideas would be, as you guys have heard, right, there's this belief that sometimes uranium is, let's say, excreted rapidly, but I think that an exciting pathway, and also I forgot to say, I dedicate this talk to my good, well-remembered Scott Burchell, right, who got us into this idea of thinking more about the, especially with these conversations with Elicio Castillo and Julian about the gut microbiome and seeing what the gut role would be on the accumulation. So I think that is a good question right now, as far as I know, and it hasn't been addressed clearly, but a great opportunity for our research community to take these results into something more meaningful to health. So I'm not sure if with this I answer that question, but it is an opportunity.
You gave me some idea, so that's.
It's an opportunity. Yes. Yes, Anirban.
I was just curious, going back to the water pH that you showed in the very first slide. So you have 3 point something to
Yeah.
9 pH.
Mhm.
So do you see, because the speciation of dissolved uranium is a little different, right?
Yes,
pH 3 uranium would be a cation plus, and
Yes.
the other one pH 9 which are carbonate anion. Did you see any
Yeah.
change in transport in those actual sites?
yes, that's an excellent question. What I can tell you is, even with uranyl arsenates, we did some very nice research in collaboration with Peter Burns at Notre Dame and Tori Forbes, where when we have acidic pHs on Irvan, if you have enough arsenic and uranium, you can actually have uranyl arsenate complexation and even precipitation up to a certain extent. And that could be a mechanism for immobilization of both at acidic pH. When you have higher pHs, as you saw, it's more challenging, right? So that's why we have this idea of hydroxyapatite, with the challenge then that you have to be aware of, as Steve asked, with the competition of uranium with carbonates or uranium with hydroxide, right? Which would also cause a slight mobilization there. So I think those are things to keep an eye on. I don't know if that answers your question.
Certainly related, some of your experiments were
Yeah.
heap stuffer or pipe stuffer, I don't remember what it was. but
Yeah.
It wasn't
With the
carbonate.
plex.
Yeah. Tried any of those experiments with carbonate stuffer?
We did, yeah. I mean, all of these, so let me see. I believe these experiments. Let's see. Is it here? Is it in the plants maybe? Is it this one?
pH with heap stuffers.
Give me 1 second. Yeah, so let me tell you what we did here in these experiments. We actually did carbonate to these solutions around 240 milligrams per liter as well, because we try to do it replicating the alkalinity that we see on the field. So all of these waters have carbonate, and it's just not included in the ions here, but they do have. I know that. And the other thing would be that HIPS was added just in case. It was not that we relied on it. Sometimes we saw some problems in certain experiments, we added just in case. Yeah. But yeah, we definitely have done all of this with carbonates too. Yeah, that's very important, because that would be a big miss, right? If we did. Yeah. Yeah. Okay, Whitney, anyone online or anything? Okay, well, with this, you know, I really thank everyone for having me. This has been so much fun, and I'm looking forward to our continued collaboration, now that we are a community. So I really appreciate the questions that I've gotten from Manish and others on looking for ideas for collaborating, right? Because we have to stay strong, you know, collaborating and looking forward to do more exciting things. Thank you.
Thank you. Let's take a picture now.
The picture.